Welcome everyone. This is a new episode where I will be discussing virology and a lot of other intriguing questions with my colleague in UCD School of Veterinary Medicine, Dr. Gerald Barry. He is a renowned virologist, and some of you might have already heard of him or even have interacted with him in different conferences. In this episode, I chatted a lot about viruses with him, and I'm sure you will be equally excited to hear my conversations with him. Sit back, relax, and enjoy the conversation that I had with him. Gerald, Dr. Gerald Barry, but Gerald to me. Mm-hmm. And I'm really honored to have you in this show.
Well, it's fantastic to be here. Thank you for the invitation.
Yeah, well, it's my honor as well, because it's kind of not strange, but kind of funny that we are colleagues in UCD School of Veterinary Medicine, but it is because of this work, and it's sometimes we don't have the conversation or this scholastic kind of sharing of ideas, which we should have done because our offices are in close vicinity, but that often doesn't happen. And now we are sitting in a studio and we are having this conversation. I know. But it's a nice feeling. So thanks a lot for coming. Okay, again.
Great to be here.
Fantastic. Now, Gerald, you have been working in the forefront of virology research, and given the pandemic, given the different zoonotic disorders that have been constantly emerging, especially in over the last decade, I would say. Mm-hmm. So there are a lot of interest in virology, and I thought there is no better candidate than you to actually catch up on an afternoon and ask few of the questions that even I have, and I'm sure our audiences also have. Mm-hmm. My first question, and that is usually I start my show with my guests, that I ask them to give them a kind of a trajectory to their career, how they evolved into the researcher that they are today, because I think there are a lot of aspirants in academia, young PhD students, postdocs. This type of cheat bits of information is very helpful, and there is no formal way of getting it. Mm-hmm. So I'll stop now and I will let you speak out about what does it take to make a Dr. Gerald Barry.
Well, I mean, the first thing I would say is I wouldn't quite say I've stumbled into my career, but I started off with very little planning. I would readily admit that. I started doing a degree in genetics in Trinity College. I was keen on research, didn't particularly know what area of research I was going to end up in, but I knew I wanted to continue in research, and so I knew if I was going to do that, I needed to do a PhD.
Why did you like research?
I think I have a brain that likes to understand how things work. I like to ask questions. I like to dig into the nitty-gritty of what I'm observing. And I like the idea of generating knowledge that fills in the gaps that I see. Mm-hmm. And research gives me that opportunity. Probably one of the most enjoyable things about my job is intellectual freedom to explore and try to answer questions that come up on a day-to-day basis. And
Agreed.
That to me is the beauty of research. Mm-hmm. And I started to kind of evolve that, I suppose, in university, or maybe even before that. And so I went to Edinburgh to do my PhD, and I again didn't really know that I was going to end up in virology, but I was interested generally in cell biology. Mm-hmm. And I was fortunate enough to meet a professor called John Fazakerley, who was based in Edinburgh University, and he was a virologist who was also very interested in cell biology and how viruses were interacting with cells. So it was kind of a little bit of the nitty-gritty. We were getting into the weeds of what was really going on at a cellular level when a virus was infecting an animal, an insect, a human, whatever it might be. And that was really kind of what intrigued me. I knew I was interested in cell biology, and we agreed, I suppose, through a number of conversations to use viruses as a tool to try and explore the inner workings of the cell and what was going on there. And even at the beginning of my PhD, I would readily admit that it wasn't necessarily the virus that attracted me. It was more that understanding of the cell, and we were going to use the virus as a tool to explore that. But I did my PhD for 3.5 years, and I would say over that time, my general interest in virology increased. Of course, I was doing a lot of reading as I was doing my research. And there was a number of, I suppose, events during my PhD that really kind of piqued my interest.
Okay.
I remember my supervisor coming into we had a large communal office where we had lots of PhD students working together. High stress environment, as you can imagine.
Well, I could imagine very well.
Yes. But I remember my supervisor coming in one day and saying, have you heard about this outbreak of a virus called chikungunya virus?
Chikungunya virus, yes.
Which is a virus in the same family as the virus we were working on for my PhD. And I hadn't heard of it, but of course my supervisor, as he was always ahead of the game on these kind of things, and he told me about it, and it just struck me that this was a viral outbreak that was very closely related to the virus that I was studying, and it was causing disease in people, and it was new and novel, and it was of course traumatic for the people that were being infected. But from a research point of view, it was fascinating because it was something new.
Something novel, you mean?
Yeah, novel. It had circulated and caused low levels of infection, probably for millennia. But it suddenly started to move and cause disease in much larger numbers of people. And there was a lot of intrigue in that from my point of view, as to understand what has happened here, what's going on. Can we understand what the virus is doing or why it's doing it that has suddenly changed its pattern of spread? And that was probably one of the first times it really struck me that the kind of research I was doing was really directly related to a disease, an outbreak, a major event that could affect a large number of people. And of course, as virology has evolved probably over the last 10 years, those kind of events, unfortunately, are becoming more frequent, and that interest in that side of virology has really grown over time.
Now, that's great. So in fact, it's nice too, actually, that I have even chatted to you, that we both are connected to the great city of Edinburgh, isn't it?
Yes, of course, yeah.
Because I did my MSc thesis work and a lot of research that time, growing up in my earlier days in Edinburgh. It is a fantastic kind of a
city.
Triangle and I think it had a great research culture there.
Yes, absolutely.
It had a very old kind of a heritage, and all the universities working at that time, at least, with a lot of collaboration going on, a lot of exchange of ideas.
Yeah.
It was a kind of a melting pot for new ideas, and in fact, Edinburgh has a lot of role to play if I am in research now. So I think we both are very grateful to the Scottish capital, isn't it?
Yeah, absolutely. Yeah, absolutely.
It was fascinating.
Yeah, where I worked, at least for the latter part of my research, was based in the Roslin Institute.
Roslin Institute,
of course, famous for Dolly the sheep.
Yes.
The first cloned animal, and that was really the kind of bedrock of a lot of kind of animal research, I suppose. But you're right. Scotland in general, and the UK in general, actually, is a wonderful place to do research.
I can agree on that.
The environment is fantastic, and in Scotland there's some wonderful institutes, Edinburgh, Glasgow, of course, St Andrews, wonderful places, and collaboration is key.
Yeah.
And it's a small community there, so people talk and engage with each other, and it's a really fantastic place.
It was a fantastic place, although some people might not like the weather always, but
true.
Once you are inside a laboratory, I think things get better.
Yeah, it's always warm in a lab.
Warm in the lab. You are absolutely right. Now, as I said, virology has become kind of, again, into the spotlight pretty much. We are just out of a pandemic, and I'm using the word out of pandemic in a very conservative way. I don't know how out we are. And again, that pandemic phase was quite stressful, I would say. Now, regularly there have been outputs from you in Irish media and otherwhere where you have expressed a lot about COVID-19. That pandemic happened, and probably somewhere it is still ongoing in our society. I have a couple of questions which will be not like almost a traditional question, because a lot have been discussed about COVID-19. My first question to you is, we are happy we are out of this very stressful lockdowns and sometimes really draconian lockdowns where we just got into a prison. Our homes became a prison, actually, in de facto. What are the lessons of this pandemic?
I think the first lesson is these events can happen. And to me, that's a key lesson, because I think prior to COVID-19, I don't think it was fully appreciated by the general public, but probably more importantly, decision makers nationally, globally, outside of perhaps the medical sphere, that such an event was really possible on the scale that we witnessed in COVID-19.
Is this complacency that this can happen, this will not happen? Is this because that we invented quite a bit of antiviral drugs or antibacterials? We have made a lot of advancements and we thought we are invincible?
I think there's probably an element of that, yeah. I think when you look at something like the Spanish flu outbreak of 1918, and you look at how the world responded to that, and you look at some of the historical images as to what people did, and you see mass graves, you see mass rooms full of hospital beds, you see people wearing masks, you see people closing down their businesses to limit contact. This was 1918.
Even deserting their homes or anything.
And then you project forward to 2020, and in a way we reacted the same way. So one could argue we haven't really advanced that much. Now, of course, in terms of development of vaccines and stuff, we have, but that initial reaction hasn't really changed that much. And I think perhaps there was a complacency. We live in an era now where we have incredible medicine, incredible antibiotics, that fear of a pathogen causing mass illness or death perhaps has left us a little bit, because we live in an era where that kind of event is so unusual. In the past, it wouldn't have been unusual. So yeah, maybe there was an element of complacency, maybe there was an element of we can cope with whatever is thrown at us. We have the skills, we have the technology to deal with these kind of things, so we don't need to worry as much. But I think if you look at the root of it really, and you look at what was being said prior to COVID-19 on a regular basis, at the level of academics in universities all the way up to the WHO. There was constant messaging that these kind of events could happen, and we needed to prepare for them. But I don't think it was acknowledged, particularly by kind of perhaps the decision makers in the world, that we needed to take it as seriously as we probably should have. And so from a lesson point of view, I would like to think that we would now take these kind of messages more seriously. Unfortunately, looking at what's happening, I don't think we are. And I think we're reverting back to the way we always were. And I still don't think there's a recognition and an acknowledgement that preparedness is a good thing. And you can spend millions on preparing for something, or spend trillions on suffering from something. And I think that, unfortunately, that penny doesn't seem to have dropped. Because a lot of the things now that I kind of assumed we would be doing, a simple thing, for example, like proper surveillance of emerging diseases. You mentioned zoonotic infections. Yes. Proper surveillance. There's a lot of countries have stopped their surveillance programs. A lot of countries don't do good surveillance at all, or don't do broad enough surveillance. Simple things like that, okay, there will be a cost associated, but it's almost like an insurance policy whereby you spend a little bit now and it insures you hope, against the much bigger event that could come.
Yeah.
So there's a kind of a dynamic there that I'm not quite convinced by. So I think that's a key lesson that I wish, I suppose, we would learn. I think the other key lesson really for me is that understanding viruses, understanding how they cause disease, understanding how they emerge, they're fundamentals to building a protection against
protection shield, you can state,
a shield against these kind of events in the future. And so it's critical in my mind to really know what we're dealing with, know what's out there, understand the dynamics of how viruses emerge, how they jump from animals into humans, for example. What is it that as humans we are doing to encourage or allow those jumping events? So, you're thinking about things like encroachment into areas of the world that perhaps exposes us to viruses or to animals that are carrying viruses that we really shouldn't be exposing ourselves to in the rapid way that we are. I'm thinking of places like the Amazon jungle, for example, where deforestation is leading to barrier breakdown that would naturally limit our exposure to viruses.
So we are doing nothing. Humans are doing a lot of things, actually.
I think we're doing a lot of, if you were to write a script as to how we would encourage zoonotic disease to move into and cause outbreaks of disease in humans, I think a lot of what we're doing as a world would be on that script. Things
so we are
like
doing great on the verge of self-peril.
Yeah, in a way. I think we're doing some things that are not sensible from a zoonotic disease control point of view. Absolutely.
And more viruses will come over, and our unpreparedness is
because
there.
The challenge with viruses in general is that they're really good at finding niches, and they're really good at adapting to new niches. And if you continue to provide an opportunity for a virus to move into a new niche, then eventually many viruses will figure out a way of doing that. And so, if you continuously expose a virus to a new population, be that humans or whatever, every now and then the virus is going to strike on a formula that allows it to suddenly move into that human population and spread in that human population. A lot of viruses are very stable, but there are some viruses that evolve rapidly. They mutate, they change, they test the water, and they try to figure out a new formula to because if you think about it, humans are a wonderful population of animals in the world to infect. There's lots of us. We live in very close proximity to each other in most parts of the world, and if a virus can crack that and get into the human population and figure out a way of spreading, as we saw with COVID, there's a lot of people to spread into. And a lot of those won't necessarily cause major pandemics like COVID-19. But again, you can't rule out that possibility. And every now and then that kind of event will happen. And so we need to, I think, understand what is circulating in the world, what has the potential to move into humans, what has the potential to spread in the human population, and how can we, I suppose, create both kind of medical barriers, perhaps through antivirals or vaccines, but also how can we change the way we're living and interacting with the environment, animals, to create a better balance that limits those opportunities for viruses to move in. I think that's a very important lesson that, again, I'm not sure we're quite learning. And it's challenging, right? Because there's economic pressures, there's population pressures to expand, to develop, etc. Different parts of the world have different dynamics to deal with. So these kind of things are, they're very, very challenging issues. But globally, these are the kind of things, and it almost kind of sounds a bit like dealing with climate change or something, because these major challenges have potentially big impacts on how we live. But they're kind of important questions that I think we need to be thinking about if we don't want to go through what we went through, over the last few years.
That's the lesson. But then what I have understood also in my life, and I'm of course getting enlightened by you, that viruses are extremely polygamous, extremely manipulative. They can manipulate a foreign immune system, but at the same time they can manipulate on their own level. It can really play around different games. And once again, it's my view and only that viruses probably present a very different challenge than bacterial diseases. My question is why? I mean, they seem to be behaving quite differently to many other microbial agents. What is the origin of the virus? I mean, how did virus, are they terrestrial, extraterrestrial? And I know many of these questions are unsettled and we don't know because it is not possible always to give a definitive answer, tracing back evolution, tracing back life. And is virus can be considered to be life, a live entity or... These are very difficult questions, and I will not go to a lot of things into this podcast because that becomes something else. But just a simple thing, what you think has happened, or what is also discussed in textbooks might be that what is the current research consensus on the emergence of virus on our planet? Any theory you want to propose?
I don't think there is a consensus. I think that's probably the first thing to say. It's very, there are theories, I guess, as to the origin. I mean, in their essence, viruses are very simple things. They're, a bit of protein,
that
basically
is even,
wrapped around
that is even surprising.
a bit of
that
nucleic acid.
Complex, yeah.
No, they're generally very simple things, and so simple that if they don't manage to get into a cell, be that a human cell or an animal cell, let's say, they're effectively like pieces of dust. They don't do anything. They're completely reliant on a host to provide extra proteins that will allow them to replicate and make copies of themselves. So in their essence, they're very, very simple things. And I suppose you could imagine how that might occur evolutionarily speaking. These kind of simple things could have been potentially some of the first things that emerged by chance, where protein has just perhaps enveloped nucleic acid for some reason, or has broken away from a more complex cell perhaps, and figured out a way to persist. But it's all these kind of chicken and egg kind of scenarios, right? And figuring out exactly how these kind of things emerge. I'm not sure we ever quite will understand exactly what came first, where they came from. But I think ultimately, and it's why I used the word admiration earlier on, that, you kind of have to admire things that are so simple, so small that you need an electron microscope to see them.
And still can have such an impact.
And can still have such an impact. Yeah.
That's
simple, but actually so complex. And to me, that's the kind of the interesting thing about viruses. But and it's that simplicity in a way that differentiates them from things like bacteria, in that they are generally much more simple, in inverted commas, than something like a bacteria. They are completely reliant on a host to allow them to make copies of themselves, and even how they kind of persist and evolve and change over time. There's a huge dynamism to a lot of viruses. That evolutionary process is constantly happening, where millions of new particles get formed every day, many of which are completely useless because they have errors or mistakes. But slowly but surely the population kind of increases and changes and evolves. It's interesting when you look at, in thinking about the origins, we have viruses out there. Of course, we think about viruses that infect humans or animals. But of course, there are viruses that are specifically infecting bacteria, for example.
Bacteriophages.
So even at that kind of simple level, we have those kind of interactions. So it's likely that as soon as a simple unicellular organism evolved, a fraction of that figured out a way of interacting with it. And on a basic level, perhaps that's how viruses have come to be, in that they've just figured out ways of interacting, stealing from perhaps unicellular organisms in a much simpler way, but living, in inverted commas, with these organisms and kind of evolving with them over time.
I think even plants have a lot of viruses.
Absolutely.
It's a completely
you can think
domain.
of pretty much anything has a virus that infects it.
I might have I should not say stupid question, but just an inquisitiveness on my side. Can plant viruses jump to humans?
Well, that's a good question. Generally, we wouldn't think about viruses from plants that jump into humans. They tend to be quite specialized. The structure of a plant cell is very different from a mammalian cell.
Yeah, they have a cell wall and everything.
Right, exactly. And so they tend to be quite adapted to plants. I can't think off the top of my head of a plant virus that can jump to humans. There may well be one. Perhaps a listener can write in and tell us.
There's always one, isn't it?
But that wouldn't be something we would generally be particularly concerned with.
Yeah.
It tends to be more coming from an insect or mammalian origin rather than a plant.
Given the circumstances that we are going, I know I would like to ask a couple of questions on the antiviral drugs that are emerging. I know for case of antibacterials, there is a lot of resistance reported. And that's, I think this is a major topic on a global scale. Again, probably we are a bit more complacent, and we should have been more proactive, taking a pragmatic role in handling this. But then again, there are other issues which are unfortunately prioritized sometimes, but that's okay. Now, how much is the resistance a problem? Is it a problem? And if yes, how much of it is a problem in treating viral diseases?
It's a major challenge. As we mentioned earlier, the evolution of a virus is something that is constantly happening. And the virus will evolve to get away from the immune system, for example, or to improve its ability to transmit. But one of the other reasons, or one of the other triggers for evolution of a virus, are the use of antivirals. Probably the best example is HIV in humans. There is a range of antivirals that are used now against HIV because when the first antivirals started to emerge in the 90s, it was found that very rapidly, if you treated somebody with a single antiviral, although initially it would have an effect, within months, the virus circulating in that person in many cases would have evolved, or in that population that the drug was being used, would have evolved a resistance to that drug. The way we get around that with HIV now is that we use a panel of drugs all at the same time.
Yeah, so a regime.
Exactly.
Yeah.
So typically 4 or 5 drugs all together, such that if the virus evolves a resistance against one, the other 4 kick in and kill it or inactivate it. And so you're kind of constantly battling against that. We see hepatitis C is another nice example. There are some amazing antivirals for hepatitis C now. But already we're picking up on populations that have resistance to these drugs that have only really been on the market for a small number of years. And you see the same thing with SARS-CoV-2, for example. Yeah. The antivirals that are being used in, granted, a small number of cases against SARS-CoV-2, there is resistance being picked up in that virus as well. Yeah, so antivirals tend to be very, well, let's say antibiotics tend to be quite broad against bacteria. So you can treat using one antibiotic a number of different types of bacteria. Antivirals in general tend to be quite virus specific. And one of the reasons for that is they tend to target specific mechanisms about a particular virus. Some are a bit broader and a bit more generic, but in general, they tend to be quite virus specific. And what that means, of course, is that if the virus can change even just a little bit, the drug that used to perhaps plug into a viral protein and block its activity loses its efficacy.
Because of receptor binding or something?
Could be, it depends on the mechanism. So some antivirals will try and stop the polymerase, for example, so how the virus replicates its genome. And there needs to be an interaction there, perhaps directly with the polymerase. And if that polymerase changes its shape a little bit, then the drug may not be able to interact effectively. And so that slight change in shape may still allow the polymerase to function, but the interaction with the drug is lost, and so the efficacy of the drug is lost. And so, you know, I think it's an important concept with viruses is that, you know, we think about when we get infected by a virus, typically in your mind, you might think, okay, I've got a virus, it's like one thing. But actually, when you're infected by a virus, you're infected by a cloud.
Yeah,
it's
trillions, trillions of particles.
Exactly. And within that cloud, there are variants circulating. And the dynamics of that cloud change constantly. And so within that cloud, there may be a version of the virus that suddenly the drug doesn't work against. And so if you're infected and 99.9% of the particles that infect you, the drug is effective against.
But that 0.1% remains.
But that 0.1 then starts to kick off in your body, and then suddenly that guy gets out into the next person, and the next person, and that's how you get these resistance. So it's a huge challenge. AMR, antimicrobial resistance in bacteria, in parasites, these are growing and already now causing huge problems globally. Yeah. And antivirals are a huge challenge as well. And it's a kind of a constant race to continuously develop new antivirals that will keep pace with the evolution of the viruses that are circulating. It's very hard to stop these viruses changing. So you have to just try and keep pace with them a lot of the time with these antivirals, or as we've seen with COVID, new versions of the vaccine that try and keep pace with new variants emerging, for example.
And that's a good point, because there is a continuous race. We change, they change, then we change, then they change. And unfortunately, they have no lack of funding of research or anything. They just do it. But there are certain realities in our way of preparing new drugs. I have a quick something that have always intrigued me. If I look into antibacterials or antifungals, and till my medical college entry days in 1997, I have always seen that textbooks are getting updated every 3-4 years with new generations of antibacterials being added. I know that is also a race, and the new addition of antibacterials to this library, it's getting sluggish because of a lot of things, but still new antibacterials are emerging. But if you look into this entire catalog of antivirals, related to other antimicrobial agents, antivirals look a bit slow, number one. And number two, do you think in future fighting antiviral diseases, do you think there will be a more kind of a paradigm shift to handle it with the vaccines rather than antivirals? Or do you think antivirals will always, kind of hold on its to a ground as a kind of a frontline defense against antiviral diseases?
It seems at the moment, if we look at, I suppose, the antivirals that are out there at the moment, some antivirals are used as a secondary line. Vaccine is first. But with something like HIV, where it's very challenging to develop a vaccine, antivirals are frontline. So it becomes almost very virus specific. And in a way, that's one of the economic challenges for a pharmaceutical company to develop. And of course, they love them or hate them. They are the drivers of the development of these kinds of drugs. If you have a major outbreak of disease, such as COVID-19, there are millions of people potentially that will buy your antiviral. And so there's an economic benefit to spending millions and in some cases billions on developing a new antiviral. If you're trying to target something that perhaps isn't causing as much disease, and you have to spend the equivalent amount of money to develop an antiviral for that particular virus, then the economic benefit becomes a bigger question. And so a lot of pharmaceutical companies are perhaps less inclined to spend that money because the reward perhaps will be less. Antimicrobials tend to be against bacteria are a little bit different because, as I mentioned, they in a lot of cases are more generic. So you develop one drug, you potentially hit
you can target
many bacteria, many different infections. It isn't quite like that, unfortunately, with viruses. We haven't quite figured out a broad range of generic drugs that are antiviral against a host of different viruses. There are some, but not on the scale of antibiotics. So the economics and the dynamics there are different. And so that's why you see a lot less, I think, really effective antivirals. HIV, of course, it's a global pandemic. It continues to infect thousands, millions of people.
And it's a zoonotic disease as well. It came from...
Originally, it came from... So, there is a clear market for that. And of course, HIV is such a challenging virus because once you're infected by it, it's very hard to clear from your body. So you're on your drug regime for life. Now, thankfully, the drugs are so good now that you can live a perfectly healthy life if you have access to those drugs and you take them consistently. But and I suppose influenza, hepatitis C, there are other drugs that that have been developed for those. But beyond that, it is challenging, and I think vaccine becomes the front line for a lot of, because vaccines, perhaps the technology you don't need to reinvent the wheel every time. In a lot of cases, vaccines can be developed relatively quickly and are consistently effective. Of course, there are exceptions to that, but the development
a little bit of a tweaking of the recipe, you can target the new variant that is emerging.
Potentially, exactly, yeah.
And I think vaccines have definitely shown us that they can save a lot of lives in the COVID-19 pandemic, for example.
Absolutely, yeah. Yeah, I think one other thing, I suppose, one other point to make, which I think is relevant here as well, is that with a lot of viral infections, and we saw it with COVID-19, the efficacy of antivirals in some cases depends on timing of administration. And so what I mean by that is if you take COVID-19 as the example, perhaps SARS-CoV-2, severe disease and death in a lot of cases was partially associated with the virus, but as the infection continued in those that became severely ill, actually the immune response became the major factor involved. So in other words, what happened in a lot of people was you got this massive immune response in the body in response to the virus, and that started to damage and cause problems in the body.
So there was an immunological storm, something like that.
Cytokine storm kicked in. That started to cause tissue damage. And it was found that if you administered antivirals while a person was in that state,
Convalescing or something, yeah.
the impact was very, very small. And in a lot of cases wasn't enough to save the person's life or to really help them recover. Whereas if you administered the antiviral very, very early in the infection, it was very effective because at that point, it was the virus that was causing the problem, not necessarily the immune response. I think bacterial infections are a little bit different. There are similarities, but antibiotics, they can really hit bacteria so hard, even in the later stages of infection, and can be very, very effective. So, the dynamics again are different with viruses and bacteria because of this immune response effect in some of these kind of virus infections, like respiratory viruses, like influenza, for example, and SARS-CoV-2. So you have that dynamic as well. So even if you have an antiviral that works, timing is important as well. If you don't get in there early enough, sometimes it can be too late to be effective.
Now, there are some viruses I know, and that can cause cancer also.
Yes.
Yes. What's the mechanism? I mean, do they put their genetic materials inside the cells? I mean,
yeah. So it's interesting. There are different viruses do different things. Probably one of the best known viruses that causes disease is human papillomavirus, HPV. Yeah. It's a major cause of cervical cancer in the population. Thankfully, we have a very effective vaccine against it now, and the hope would be that cases continue to reduce if vaccine rates remain high. And the way that virus typically causes cancer is that when it replicates in the cell, it has a DNA genome. So its nucleic acid is DNA. And that DNA has the ability to effectively cut open the cell's genome and insert its own genome
so it kind of gets intercalated into it.
It integrates and knits itself into the genome of the host cell. Of course, the benefit for the virus is once it's integrated, it is protected from the immune system. It can't.
It's
the
part
immune
of that,
system
part
in a
of
lot
the body's system now.
Effectively, and as the cells divide, it also makes copies of the virus's genome. So it kind of hides inside the cell. With HPV, one of the challenges is when that integration occurs. In a lot of cases, it triggers the upregulation of viral proteins that are pro-oncogenic. And those pro-oncogenic proteins suppress our natural defense inside the cell that would typically stop oncogenesis occurring. So proteins like P53, for example, get suppressed by viral proteins. Now, there are other dynamics happening, but this is one of the kind of key steps. The other thing that can happen is, and we see it, for example, with some retroviruses, feline leukemia virus is perhaps a nice example of it in cats, where this integration process can randomly occur. And because it's randomly occurring, that kind of cutting of the host genome, integration of the viral genome can disrupt genes that would normally express proteins that would be suppressors of oncogenesis. And so if you get disruption of those natural proteins, again, it can encourage those infected cells to transform into tumors or the replication or division can go out of control. So that kind of because naturally cells have these kind of checkpoints where if there's any errors, proteins are expressed to perhaps trigger apoptosis if the cell is not replicating.
In the cell cycle, in G1, ASG2, all these phases,
And if those checkpoints are disrupted, then you can get cells dividing out of control, and that's where that can lead to tumor formation, for example. And so, in some cases, that kind of integration of the viral genome into the host genome can disrupt those checkpoint proteins, which can lead then to the cell, as I say, dividing out of control and can lead to some cancers.
So that means in that case that HPV vaccine that will also, I mean, it's effective, very effective, but it can also stop this integration of viral genome into the host genome. And in long term, it can also give protection against cervical cancer.
Exactly. Yeah. So one of the key indicators of the potential for development of cervical cancer is the presence of HPV. And so one of the key tests now is looking for the presence of that virus. And we know there are different versions of HPV, and some are more inclined to cause cervical cancer than others, which is why if the vaccine can effectively protect the body against infection from that virus, you're much less likely to develop cervical cancer.
I mean, these are incredible information. Do we have, I know we have our viral reference labs. In US, they have CDC. In Ireland, fortunately, it is in UCD. What kind of information? I mean, are you in collaboration with our Irish reference lab, or what kind of, I'm sure it needs a lot of research, and these are risky virus, so you need really high level of protection in the labs.
Sure.
So what kind of facilities are you availing or are available to researchers in Ireland, for example?
Yeah, UCD is very fortunate. We have some of the I suppose, speaking for myself, and of course I work for UCD, but I think we have some of the best facilities in Ireland that allow us to study viruses that cause major disease in human populations. There are different categories of lab, categories 1 to 4 in terms of their containment. Containment level 4 you would work with things like Ebola, for example. Very, very lethal, likely to cause death in people. We don't have a category 4 lab. We don't have one in Ireland, but we do have category 3 labs, and 2 of those are based in UCD. The National Virus Reference Lab has one lab in collaboration with some other centres in UCD, and then the School of Veterinary Medicine, where you and I are based, has a containment level 3 laboratory as well. And that allows us and the National Virus Reference Lab and some other groups within UCD to study and understand viruses that have the capability to cause disease in human populations, such as SARS-CoV-2, for example. So we work on viruses like that. There's a group, Nicola Fletcher works on hepatitis E, for example. Stephen Gordon works on TB, tuberculosis, which is a bacteria course, but also that high containment. And the National Virus Reference Lab then has to have access to a lab like that in case they ever come across a case of a virus perhaps they can't readily identify and need to be able to study it in further detail. Having access to that high containment lab allows them to do that. And of course, that was crucial during the COVID-19 pandemic. When we consistently collaborated with groups from all over Ireland and internationally, perhaps that didn't have access to labs like that, and we opened our doors and readily collaborated with people and trained extra people to work in these kind of high containment environments.
Now, in fact, it's interesting because we are based in School of Veterinary Medicine, of course, and a lot of these viruses, in fact, almost all of them are coming from animals.
Yes, a lot of them.
And you know, we both have been very much involved with One Health concept.
Yes.
This is, again, my observation and speaking only on my behalf, that what the world in 2023 shows that there has to be an integrated One Health-based approach where human medicine has to have more crosstalks with veterinary medicine. Because I think if it is coming from an animal, we have to know the animal, what it does in animal. And I'm sure there are a lot of information floating out in the veterinary medicine domain which can actually help us.
Absolutely.
That's One Health approach, and we both have worked quite a lot on that. Strong proponent of One Health.
Yes.
But I still don't see that much of integration of, medicine, human medicine and veterinary medicine coming together sometimes. What you would say to encourage this kind of collaboration or being like traveling it together?
Well, I think it's a huge challenge. Even now, I think there's perhaps a lack of recognition of the need for those cross-discipline interactions and engagements. I always give the example of rabies. And I would say to, groups of students perhaps, how do you control or limit the infection of people with rabies? Different groups, depending on their background, will have different opinions. And the best way to stop people dying of rabies is to stop it getting into humans in the first place. So you talk to your local vet and you say, how can we vaccinate all the dogs against rabies? Because 99% of human infections come from dogs.
Yeah,
So
and they're
if
100% fatal almost.
rabies is incredibly lethal, one of the most lethal viruses out there if you don't get access to treatment. So that is a perfect example where human medicine and veterinary medicine have to engage. And of course, it goes beyond that into the environment and engaging with perhaps bats and wildlife as well. I think, and I see it more and more, that interdisciplinary approaches to dealing with major problems is the only way we're going to solve them.
Yeah.
And it really is about bringing people together that look at perhaps the same problems, but from different points of view.
Yeah.
And you only really do that when you come from different disciplines, because, and I see it all the time, when somebody is in veterinary medicine or human medicine or science or whatever it might be, and they're constantly thinking in that space, then they do become a little blinded to other possibilities, other approaches, other ways of looking at things. But when you put people together, suddenly those ideas become obvious and readily available to everyone.
Yeah.
So the One Health approach would encourage the idea that in order to tackle any major challenge, we have to consider it from not only a human point of view or an animal point of view, we have to consider both, and we have to consider the environment as well. And you know, when we talk about, I suppose, viruses and zoonotic infections, you know, how do you stop a zoonotic infection moving into humans? It isn't good enough just to wait for it to happen and figure out a cure. You have to think about, okay, well, where might it come from? It might come from an animal. So can we understand what's happening in the animals? Can we protect the animals from getting the infection in the first place? Where are those animals getting it? Is it circulating in insects, for example? But also then beyond that, how is the environment contributing to that transmission? Is there something about, as I say, people engaging with that, people moving? What are the dynamics of the population? So multiple layers of understanding a disease allows you to properly tackle it, you know, and I think the key thing is trying to encourage that recognition. Because from my own experience, and we've recently established a One Health Centre in UCD that officially kicked off yesterday, in fact.
Congratulations.
One of the key points of that centre is going to be about identifying and bringing people together from different disciplines and encouraging them to look at problems from different angles, encouraging them to consider the broader outlook on something. I saw a very interesting example of it that I thought was really nice and illustrated the point recently. There was a case of farms in the east coast of Ireland, and they were cattle farms, and they were seeing big jumps in cases of bovine TB. They couldn't quite understand why this was happening. And where the farms were located, on one side there was a beach, on the other side was a forest. And so for years they had worried about badger incursions, because we know that badgers can potentially transmit TB to the cattle. And that was a kind of an ongoing issue. But they saw this sudden spike and wanted to understand what that was. There was an investigation. It turned out that there was houses being built, maybe half a mile down the road, that incurred into the forest, knocked down some of the forest, damaging some of the habitat there, but also creating a lot of noise, disruption, and it caused an influx of badgers into the farms to get away from the human encroachment in their habitat. And with that came TB. And so when you look at that as a dynamic, from a veterinary point of view, you might think just about the cows. From an environmental point of view, you start to think much bigger than that. And you start to think about habitat as a whole and human interaction with that habitat, and how one activity can impact and have unintended consequences on lots of others. So that One Health approach really encourages that much broader approach to trying to tackle a lot of these things. And by doing that, you hope that you solve those problems in a much more effective way.
Could we have done things better, or could we have prepared better in future also, even for COVID-19? Was there some knowledge already out in veterinary domain that probably we could have used immediately if such collaboration would have been already in place?
I think it's interesting when you look at coronaviruses. I mean, we can answer that in a number of different ways, I suppose. We could talk about the origins of it and trying to stop it moving into humans in the first place. if you believe the idea and the origins of SARS-CoV-2 are still up for debate. But if we take one suggestion, which is it came from farmed wild animals in a market in Wuhan. If that's the case, we saw a very similar situation about 10 years, 15 years previous when we had SARS-1 outbreak. Similar kind of origins. So if we had learnt anything from that, we had learnt that the idea of farming and bringing live wild animals into close contact consistently with humans is perhaps something we need to control better, eliminate, or at least put controls in place to stop the potential spread of a virus from one to the other. If we look at it, I suppose, from a different point of view, and we think about could we maybe have approached or reacted perhaps differently? One example I always think of when I think about coronaviruses in general, and I think about coronaviruses that circulate in animals, and respiratory viruses in general in animals. One of the key things for me is the idea that you can get commensal infection of a virus, which basically means that healthy animals and humans can carry viruses and not have any disease. And other factors such as stress can lead to an exacerbation of that and disease subsequently. Bovine respiratory disease is a nice example of that. If you have a nice, happy, healthy cow, they can carry some of the viruses associated with respiratory disease all day long and they're unlikely to get sick. You put them under a stress and suddenly you'll start to see sickness.
Is it because of immunological fluctuation?
Yeah, environment changes, the immune system responds to it. And you see the same in humans. Stress can lead to cold sores emerging, for example. So if you take that into account, what that also means is you can get spread of viruses independent of clinical signs or independent of symptoms. Now, a key learning within that is, if we project back to COVID-19, one of our first actions in early pandemic times was to take older people out of hospitals. The sensible approach, because where are people going when they have COVID? They're going to hospitals. So let's get older people out. They're vulnerable. We don't want them to be exposed to COVID. We're going to put them back into nursing homes or retirement homes or wherever they were going. And there was an assumption made that if they didn't have symptoms, they couldn't transmit the virus. And that was based on knowledge we had learned from SARS-1. And we know now that with SARS-1, symptoms seemed to be associated with transmission. So the assumption was made if they were happy and healthy and didn't have symptoms, they couldn't transmit the virus. And so putting them back into a nursing home didn't require testing. We would base it completely on symptoms. Of course, the fatal error was that we now know that SARS-CoV-2 can transmit very happily from one to another, independent of symptoms, and is in fact very effective the day before symptoms emerge, particularly. And so what happened, of course, was a lot of older people went into nursing homes or retirement homes or care homes infected. Nobody knew they were infected, and that led to huge outbreaks or contributed at least to huge outbreaks. The learning from animals there is that, and what most vets would readily tell you is that this is an issue. Spread independent of symptoms is an issue. And I know I've spoken to a number of colleagues that were involved in kind of, the kind of decision-making process around these kind of things. And they would say that one of the errors they made was that they didn't acknowledge or consider that spread could happen independent of symptoms.
Even in asymptomatic cases.
Exactly. And so that is a dynamic, I think, that was a major flaw. And if people had a broader...
more holistic idea.
Exactly, that would have been recognized and would have been considered. So those kind of lessons, when you have people talking to each other, I think those kind of lessons would have been learned a lot quicker.
I completely agree. Are there viruses now, Gerald, that worries you, that you think are
the next one
doing conspiracy? We don't have an extra, but just one or two you think, or you can point out one or two just that you think you will keep an eye?
I think the one on everyone's lips at the moment is avian influenza. Influenza in general is one of these viruses that can change very rapidly. and new versions of it can emerge, consistently emerge, and every now and then a new version comes out that starts to cause a lot of a disease. We've had multiple pandemics in the past with influenza. What's going on with avian influenza is interesting at the moment. So this is a version of influenza that circulates predominantly in bird populations.
Yeah.
We see outbreaks on poultry farms sometimes, but we're consistently now seeing a kind of a change in pattern, I would say. In Ireland, for example, typically avian influenza would have only been seen traditionally in the winter. We're now seeing cases fairly regularly, even during the summer, suggesting that it has gone from something that comes in and goes away to something that's with us all the time. What we're also seeing globally as well is major outbreaks of avian influenza in animals that are not birds. So, for example, 5% of sea lions in Peru, I believe it was, were killed by an avian influenza outbreak. So 5% of their total population. So you're seeing outbreaks of avian influenza in other animals apart from birds.
So they're changing hosts.
Yeah,
and there's a pattern.
That kind of pattern of big outbreaks seem to be occurring. Maybe we're detecting them more effectively, granted, but they seem to be occurring more regularly. So, and of course, we're interacting constantly with birds that are carrying these kind of things. So that's, I think, something on everyone's watch list, definitely. And then, of course, I think you mentioned it right at the beginning, pathogen X.
Pathogen X.
You know, if you were to ask somebody 6 months before COVID-19, what's going to be the next big thing? Coronavirus was fairly low down the list.
Yeah, but then
it became the next big thing. So one of the major challenges is there may well be something that comes out of the woodwork that we just don't expect. And that's when you get a big problem, because if influenza comes along, we have antivirals, we know how to make vaccines. We can probably very rapidly roll those out in a population. And so you would hope we wouldn't have on the scale of a COVID-19 type pandemic with influenza, even though it may well cause some problems. If something comes along where we don't have an antiviral, where we don't have a vaccine ready to go, then you get a COVID-19 problem. And I think although COVID-19 caused, huge problems, of course, and tragically killed a lot of people, in hindsight, actually, I firmly believe that we dodged a major bullet with COVID-19. Because if you look at the pattern of disease, young kids weren't really that badly affected. And if you compare that to, say, Spanish flu in 1918, the predominant people that died from that were children under the age of 5 and those in their 20s. The healthy population of the world. That's a very different dynamic if you think about it. Imagine a scenario where we had COVID-19, but those that were dying were the under 5s. That creates a much bigger thing. And generally the mortality rate of COVID is very low across the whole population. But you take something like MERS that has a mortality rate of 20 to 30%. 1 in 3 people get it, die.
Then there is a problem.
That's a big problem. So, I'm not trying to scare anyone, of course, but the realism is that a virus can emerge and it's difficult to predict what it's going to do. And so it's about, I think, putting preparations and plans in place to be able to say, if something comes out, the next thing that comes out, how do we deal with that? How do we manage that? I go back to the idea of insurance. We have to put insurance policies in place to allow us to be able to respond effectively, and acknowledge that these kind of things are consistently possible, and when it'll happen, it could be tomorrow, it could be 100 years from now. We don't know, but it's a when rather than if kind of question, because it'll definitely happen again.
It is inevitable.
Absolutely. Guaranteed.
Yeah.
We just don't know when it's going to happen.
We are in the last segment of this story. I mean, our conversation can go on for hours and hours. But the truth of the matter is that we have to end somewhere.
Yep.
I've just last couple of questions for you. I know vaccines have been effective, and one of the major achievement of vaccine is eradication of a couple of diseases. For example, smallpox has been kind of eradicated. Are there more viral diseases that you think has the potential to be completely eradicated in near future with the vaccine or something?
So smallpox has been eradicated. Thankfully, rinderpest in animals has been eradicated. There's talks that HPV could be not eradicated, but eliminated perhaps from certain parts of the world. I think one of the challenges with a lot of viruses is developing a vaccine that stops infection. COVID-19 vaccine does not stop infection, so it continues to spread. So you need a vaccine that stops infection, and you also ideally need to target a virus that doesn't have a wildlife reservoir.
Yeah.
It's very difficult to eliminate something if it has a wildlife reservoir. Smallpox was only in humans, so we knew if we got rid of it in humans, it was gone. So that's really the big challenge. Something like influenza, for example, which we would love to eradicate, that's going to be very, very difficult because you know it circulates in so many animals. I think it's a difficult one. What's next on the chopping block? In terms of animals, it's easier to do. You can control it through a number of different ways, and you can regionalize things. We're eliminating bovine viral diarrhea virus from Ireland, for example, currently, and we'll be free of it hopefully by the end, if not early Q1 next year. I think in many cases there was a hope that hepatitis C might be eradicated. I think that's a possibility to move towards. I don't think it looks like it's definitely going to happen, but you know, I think these kind of things are something we can aim for. But really, I don't think we need to think about eradication. It's about control. We have to be in control. We can't let the virus be in control. And so we need to, you know, build shields that give us control. During COVID, we lost control completely.
Yeah, that's very
And we had to react very draconally, locking us all down. You know, that can't be our go-to response. It shouldn't be.
For sure.
And it became our go-to response during COVID, and it really shouldn't be. We need to move to a position where we have shields that we can put in place that are generic enough, or we have a broad enough specificity library that allows us to say, whatever comes at us, we have a defense. So, you know, one of the things I would love to see, for example, is if we just took an approach of what are the most likely 100 viruses? Let's build a vaccine against all of them. They may never come into the human population, but who cares? At least we have a vaccine in case it does.
At least we have a library to refer to.
And we can refer to it rapidly and rapidly build those shields. That's the kind of preparation that we need to be doing.
My last question, and we have discussed viruses and we have kind of nailed it as an agent which is in our surroundings, which is always doing evil things. But I know there are beneficial viruses as well. So I know it's a huge topic. Once again, any discussion on virus will take years to even go to scratch of it. But just if I give you the job to make a quick pitch, and short but effective, impactful pitch, that viruses can do a lot of bad things, but they often do sometimes they can do a little bit of good as well. And I know from a pharmaceutics perspective, people are using viruses as therapeutic vectors, for example, for delivering drugs.
Absolutely. Bacteriophage are a nice example of that.
Exactly.
As an alternative to antibiotics, for example, as a way of targeting and killing bacteria.
Exactly.
Yeah,
so they're delivering drugs. There are other helpful things.
Vectors.
So probably like many other agents hovering around in our cosmos, viruses also do some good things and have a lot of potential, for example.
By understanding them, that allows us to use them and control and build tools with them, like vaccine delivery systems, like gene therapy systems. You know, I mean, one of the vaccines against COVID used a virus as a vector to deliver a payload, essentially, that created a protection through the use of a vaccine. So, absolutely, viruses are simple things, but incredibly complex. But by understanding them, we see how we could potentially use them to understand the fundamentals of how a cell works, but also to build tools that can be used to deliver vaccines, to deliver drugs potentially, gene therapy is a real area of interest for the use of vaccines. And so those kind of approaches is how we can gain a benefit from viruses, absolutely. And of course, that's ignoring things like how bacteriophage, of course, manipulate our microbiome. And how crucial they are to controlling bacterial populations in our gut. So, we can't ignore the factors like that are hugely beneficial to us. So and it's not all bad.
It's not all bad. That's a positive note we can end. And in fact, being a researcher in nanomedicine, I often see viral particles as an opportunity because you have got ready-made nanoparticles in front of you. And I know people have even, you can clear it up. I know researchers who have used nanoparticles as doing small compartmentalized reactions in chemistry labs. So you actually create a nanoreactor.
Wow.
These are published research. So for me, it's also a land of opportunity because you have ready-made nanoparticles in front of you, which are often up to 100 nanometers. Even COVID and SARS-CoV-2, sometimes they are less than 80, sometimes around 80 nanometers to 100 nanometers. So these are ready-made nanoparticles. And so basically, with our advent and with our advancement of nanotechnology, a lot of things, including drug delivery, diagnostics, imaging, for example, I think there are a lot of opportunities which remains unexplored as well.
Yeah, I suppose viruses have had, thousands of years to perfect systems like getting through mucus barriers, for example. How do you get a nanoparticle through that mucus barrier and deliver a drug,
there are
for
incredible
example?
things to be learned from
So there's a lot out there. Yeah, it's a lot to learn still.
A lot to learn.
Keep us going for a while.
Gerald, I cannot thank you enough. I think everyone will agree that it has been a remarkably enlightening discussion.
I've really enjoyed it.
Our audience will like it. I'm pretty sure I can vouch on their behalf. And of course, I have liked it a lot. There are many things that we could not touch, and I know I'm sorry for that. But you know what? Like viruses, there are always next option. There will be something coming back next again.
For sure.
I'm sure we'll find an opportunity to sit down together and have this kind of more scholastic discussion, which I'm sure audience have a lot of interest into. Thank you enough. Thanks for coming.
Thank you, Sourav.